Roundtable on Causal Inference, Missing Data, Survival Analysis, and Clinical Trials. I think this is a great opportunity to learn a little bit about the research that our faculty do here and get some details about what biostatistics is all about. So without further ado, I'll hand it over to our faculty here to give brief introductions about themselves, kind of similar to what we gave in the room: name, title, and brief academic background or research interests or whatever you think is applicable. So Phil, we'll start with you.
So I'm Phil Boonstra. I'm an associate professor here. I've been here, well, actually, I got my degrees here. So if you sort of count the totality, I've been here since 2007. So I think I talked to some of you, or at some of you, in the master's session earlier today, right? I teach 699, our capstone class, may be relevant to this. I do research in the area of ECMO, which is like a sort of a long-term heart and lung support. There's a sort of a fairly mature ECMO research community that has a registry based here in Ann Arbor, but it's a worldwide registry. Very relevant for like COVID-19 research and stuff. So I think in terms of this, like this roundtable, it's sort of as a grab bag. Maybe I'm a little bit in each of the four categories, but not really an expert in any of them, right? And sort of a jack of all trades, but yeah.
All right, Tom.
Hi everybody, I'm Tom Braun. I've been a professor here since 1999. I did my PhD in Seattle. I also was an actuary for 4 years, and I don't know if people still consider that career. I hated it, but that's why I'm here, but I'm happy if some folks are considering other careers in biostatistics. Like Phil said, I'm considered, I guess, a clinical trials expert, but the beauty of our career is what we know how to do allows us to move around. So I worked at the dental school for many years. I was a faculty member there, not because I knew dentistry, but because I did their trials. I'm working now with quality improvement in cardiac surgeries. I'm learning a lot about cardiac surgery that I didn't know 2 years ago. But since I've been here, I've mostly been using Bayesian methods in adaptive clinical trials. So using a trial, and as it's ongoing, taking the data that we've collected to determine what we're going to do with the rest of the people coming into the trial. And as I've progressed again over time, Kelly Kidwell, who's right now in Australia on sabbatical, she and I have been doing some work in looking at rare diseases and how can we cleverly look for treatments in rare diseases, which is hard because we want lots of data and we don't have it at our disposal. So happy to talk about any or all of those.
So I'm Nick Henderson. I'm an assistant professor here at Michigan. So I got my PhD in statistics at Wisconsin, and I did a postdoc at Johns Hopkins, kind of more cancer biostatistics related. And here I've kind of focused a lot on, I guess, broadly speaking, precision medicine topics in kind of cancer. So I think my work, I don't know if I'm an expert in each of the areas, but I think certainly touches on most of the areas for this roundtable. So I definitely do a lot of survival analysis, and I think a lot of the questions at least touch on causal questions, since I do a lot of work with kind of non-clinical trial data. So that's about it. So I don't know if you
So my name is Bamboo. I'm kind of a new-ish assistant professor in the department. I joined at the beginning of last year. So I don't necessarily belong to any of those four categories, but as Tom said, this is the beauty of our profession. We can be very versatile in terms of the application areas that we actually work on. So in my case, I work a lot on infectious diseases data, vaccination data, and I also work a lot on observational healthcare data. So data that were not collected from clinical trials, but rather from hospitals, from insurance companies, thinking about in the real world how treatments actually work comparatively in complicated heterogeneous patient populations. So a lot of what I do actually do touch on missing data issues, survival analysis issues, and also causal inference problems. In terms of my background, so I got my PhD from Duke University. That's how I got converted into a Bayesian statistician. But actually in real life, I'm more of a pragmaticist, so I actually do frequentist methods as well. I just don't let my Bayesian mentors know about this.
All right, for our first question, I want to talk a little bit about breaking down the word biostatistics. There's the biology part of it, the biomedicine part of it, and then there's the statistics part of it. So I want to start with statistics first. What are some examples of how you use, adapt, create statistical methodologies in any of these areas to help you answer research questions that you're investigating? Anyone can take it, yeah.
Sure. So just very briefly as an example, I mentioned the ECMO piece. The other thing I forgot to mention the first time around was I also do like early phase clinical trial design, like Tom, and sort of it's sort of like a very classic statistical problem of that you are trying to figure out what's the safe dose that you can give to patients who are being treated for a particular disease. The idea being that you want to give as much drug as possible to them so that you can like treat the disease without giving too much that you're going to harm the patient. And so there's a very classical dose response idea underlying that. And at the same time, you don't have very much data, right? So you bring to bear like a Bayesian perspective. You're trying to incorporate sort of your assumptions that you believe are like how that curve might be shaped when you're designing the trials to enroll the few patients that you are able to enroll in the trial to like get the most use out of the information that they provide. So you sort of bring to bear what sort of shape is that dose response curve? Do you think like what shape will that look like in advance? How certain are you at the lower dose levels where you might have some more preclinical information and knowledge versus at the higher dose levels you might be less certain, so that there's sort of like varying uncertainty at different points in that dose response curve. So it's a very practical clinical question that has a very close connection to like a classical statistical problem.
So I'm actually giving a recruiting talk at Kenyon College, where my daughter goes to college in a week. So this is very fresh in my mind as I'm going to talk about this. A lot of clinical trials you probably are familiar with are randomized. So we have maybe 2 treatments usually, right? And we want to believe that all factors are equal, so we randomly decide who's going to get which treatment. We're finding in a lot of clinical trials that actually patients might have a preference, or their doctors might have a preference as to which treatment they're going to get. And we are concerned by that because if someone prefers a treatment, it might be for reasons that actually impact why that treatment might work. And so we say confounding, that's a confounder. And so we can't use those data in a typical clinical trial. And historically what we've done is we said, sorry, you have to be randomized. If you're not willing, go somewhere else. But now we're believing that because the trials are so expensive, we need those data. And we have to think of a clever way, like I think we use clever. How do I take someone who was treated with A because they wanted it and use those data with someone who got A because they were randomized to it? And like Phil said, there's a classical statistical idea of borrowing data from what is typically observational data with randomized data. There are lots of folks working in this area now. And a grad student and I are using a Bayesian method to link those 2 sets of data together, again, in a way that tells me, well, maybe a preference person is worth 1/3 of the data of a randomized person, mathematically, not quantitatively. And so how do we figure out what those weights are? How does it affect what we actually come up with? We talk about bias and efficiency. We want to be confident that we have the right answer, but we also know that there's bias. It might be slightly off. And so a grad student and I have been spending a fair amount of time trying to figure out if you have a 2-arm randomized trial and you also have those 2 arms being given to folks who have a preference, how do we use all 4 groups in the analysis of the treatments? There is a clinical trial right now in Michigan, again, looking at removal of kidney stones. And when you go in to remove a kidney stone, you pull it back out through a vessel in your body. And it's unclear of whether or not a stent should be placed in there to provide support during the surgery. It's uncomfortable. It's not pleasant after the surgery. And so some doctors believe, well, maybe we should forego that. Maybe we should just remove the kidney stone. But if we want to head-to-head compare use of stents or not, there are lots of folks who aren't willing to be in that trial because either they've had kidney stones in the past and they don't want what treatment. So this trial is amazing. I'm very excited about it because it actually has more data in the preference group than in the randomized group. And we'd like to use all those data together in a clever way. So again, I'm really excited about, one, the methods paper, but hopefully the application of this to a clinical trial. is always what I think we want as biostatisticians. The methods paper is great, but when people actually use what we do, that's why we're here.
All right, yeah, so maybe I'll just mention something that just comes to mind in terms of, I guess, some, applied work that I work on that has kind of worked together with, I guess, methods development. So I guess I've done a decent amount of work with kind of, I guess, predicting or characterizing survival using kind of machine learning methods. And I think in a lot of the applied work, there's also longitudinal information, but something I think is kind of underappreciated, especially with cancer outcomes. There's a lot of, I guess you would call dependent censoring, like some of the outcomes, at least the common ways they're censored. Basically, it just means you stop following them before they have like the event of interest, which is like often maybe progression. A lot of times that, I guess that lack of follow-up is kind of related to, it's not independent of kind of characteristics which might be related to, I guess, progression. So, some of the things that I've worked on is kind of incorporating dependent censoring into kind of survival, I guess, machine learning prediction models. And also kind of goes back to Bayesian in a way that kind of reflects the uncertainty of the, I guess, your dependent sensory model, since a lot of times that uncertainty is kind of not properly reflected, and it's a lot easier to do that in a Bayesian way. So I think that's, I don't know, that's just something that came to mind in terms of something that's like, I guess, commonly encountered with particularly like observational cancer research, as something that's kind of been related to methods work.
Can I talk about something that's not immediately biomedical related,
Absolutely.
but also statistical? Yeah, because I want to give you one example where it's not necessarily a biomedical data type of application to show that even within a biostat department, you can still work on other applications using statistical principles. So this is from a paper that I did a couple of years ago. It's about sports analytics. So this was about analyzing how teamwork actually works in the context of college basketball. So we had this very interesting data set of 24 frames of videotaping of all home games at a certain, that I can name, prestigious men's college basketball team.
Duke, man.
I'm not saying anything about that. Yeah. And so the question is, well, how do we define good teamwork? How do we know whether or not good teamwork is contributing to the success of games? So what we did over there is we think about interactions between players as social networks that evolve over time. If a player passes the ball to another player, we see it as an instantaneous edge on a changing social network. But of course, a basketball game also evolves throughout the court spatially and temporally, so we can also model it as a spatial temporal process. So these are all rather established statistical problems with a lot of statistical methodologies at our disposal. So we did this very interesting little paper about applying and adapting those techniques, trying to understand what are the latent structures of player interactions that are strong predictors of game success. So a very interesting technique we used is called latent vector model. So mapping each player to a vector in a lower dimensional space. So if some of you have learned about AI, neural networks, or even large language models, they sound very familiar to you, right? It's called embeddings. So this kind of idea originated from statistics, bringing something high dimensional to a projection in a lower dimensional space so that it becomes more tractable. So we did that technique to understand the dynamical behavior of players' interactions on the court, coupled with a spatial temporal process to describe all the events that are happening on the basketball court. So we actually brought about some data-driven evidence that if players on a team are more centralized, are more balanced in their reciprocal interactions, they are more likely to succeed in a basketball game. So just to give you one example of how broadly we can apply our statistical expertise in different kinds of applications.
And if you're looking for insight into your March Madness bracket, I think
Yeah, I guess so.
Next question, I'm going to combine a couple questions into one here. So to talk about the applications of your research, I'm curious about like what are the exciting current biological, biomedical applications of your research, and then what are some future applications that you see coming down the road that really excite you that are enhanced by biostatistics? Anyone can take that one.
Oof. Again, in the world of clinical trials, the idea of preference is something that has existed for decades but has been ignored because we didn't know what to do with it. What to do with them, I should plural it. Data is plural, by the way, folks. It's not singular. The data are. One of my peeves. And trying, again, as we moved along in this idea of data integration, electronic health records, you've heard about people probably talk about this. We're learning that the idea of simply randomized clinical trial data, it's becoming infeasible. It's too expensive. It's too hard to get people. The fascinating part of my job is working with clinicians who think every one of their patients want to be in a clinical trial, when the reality is most of them don't want to be in a clinical trial. And so we're realizing that it is hard, and it is certainly we're dealing with the fact that we're not getting a representative sample of our population when we do clinical trials. When folks don't want to be in our clinical trial because they won't be randomized, is there a way we can think about using those data? So I think we're on the cusp of making clinical trials more effective. Hopefully, most clinical trials fail, which is a disappointing thing. They're very expensive. But I'm hoping that this world of where we're talking about integrating electronic health records, if there's a way to do that in clinical trials, hopefully we can find some drugs that work that perhaps we were unable to in the past because we didn't have enough patients to study.
So just a couple months ago, a team that I work with, that I was a part of, we submitted a grant for a fairly complicated clinical trial. I guess we're sort of emphasizing the clinical trial part of this, but and it's a design called the stepped wedge design. So the basic idea is in kids with ECMO. ECMO, as I mentioned, it's this long-term heart and lung support. You're plugged into the machine, the blood is drawn out of you, pumped and oxygenated and put back into you. And it was pioneered here at Michigan in really little kids, and this trial is in pediatric ECMO. And the basic idea is that ECMO, even though it started in little kids, has been standardized for adults. A lot of the machinery and the tubes are made for adults, and then they're sort of ad hoc'd and stepped down to kids that have smaller veins. And so the idea of this trial is to standardize that circuit that I just talked about. Rather than every single the strange thing about ECMO is that it's very hospital specific, and a lot of stuff is off label, which means that it's not like approved by the FDA for longer than 6 hours, and it just has this weird sort of fact about it. And so this trial is trying to standardize the ECMO circuit for kids, and it's this design that I called stepped wedge because you can't once you introduce the treatment here, you wouldn't, especially if doctors sort of perceive that it's a good treatment, you can't randomize them in traditional sense to go back to the thing that they don't like, right? It's not too different from what Tom was talking about. And so in this step wedge design, everybody starts just doing whatever they're doing, and eventually the hospitals move over to the standardized treatment. But putting together like a whole grant like this where you've got 24 different hospitals involved, you're trying to figure out everybody's current circuit. You're trying to come up with, well, what's the right way to measure? How does this new circuit work? Because there's so many different things that can go wrong in ECMO. You can get clots in the equipment. You can get clots in the patient, which manifest as strokes or other very bad things. You can have bleeding events because they're trying to actively prevent the clots, and so then you actually get patient bleeds. So trying to bring together all of these different bad things that can happen into a measurable quantity that you can actually say, yes, this new circuit works or doesn't work. And so this is by far the biggest, sort of most expansive clinical trial that I've ever been involved in. And when you just sort of realize all of the work that goes in to a trial like this, but ultimately it's very rewarding because it's very clear what the goal is. It's to make ECMO safer for little kids that so far have sort of had to be treated like small adults rather than like different physiological beings than full-grown adults. So, yeah.
And I hope you all come away with listening to Phil talk right now. He didn't go to med school, right? I mean, but we get to be part of this. We learn the content of this. I'm not a doctor, but I've worked in bone marrow transplantation for 20 years. I know so much about that. So as a biostatistician, you don't just want to work on methods and numbers. You really become a content expert in whether it's sports or medicine or whatever. It's the cool part of what we do is you get to learn so much that maybe you didn't want to study in med school or in further classes, but it's a great experience.
I could say something quickly. I don't know if I have a specific new thing in mind, but maybe kind of a new types of data that I feel like I'm going to have to deal with. So I think essentially probably just more complex, I guess, longitudinal information that's collected over time. I mean, I'm dealing with and kind of how to incorporate that into kind of your main analyses. A lot of the data that I work with, I mean, you're recording kind of lots of information over time, kind of multifaceted, and how to incorporate that, and particularly something that's, I think, emerging is how to incorporate, I guess, features that are kind of automatically constructed, maybe from electronic health records, so maybe annotation through natural language processing or other tools, so how to kind of incorporate that into your analysis while, at least I feel like, in some way respecting the uncertainty of the thing that it generates as a kind of a latent covariate. So that's just something I feel like that's emerging that I've been thinking about.
Yeah, I'll try to be brief. So one recent application I did is about understanding patterns of HIV transmissions in sub-Saharan Africa. So sub-Saharan Africa is the region in the world where in some countries more than 40% of adults are HIV positive. So the situation is very dire. And what we did is trying to quantitatively understand what kind of population groups are at higher risk of getting infected. So we got really lucky with data collection efforts that actually collected genetic sequencing information from all the HIV positive patients. And we did, I won't go into details of the statistics, but we did a comprehensive analysis trying to understand who infected whom, what kind of population group would be responsible for infecting whom, and trying to understand the transmission pathways. And we learned that a lot of adolescent and young women between 15 to 24 years old were actually infected by men in their 40s. And then they would later on infect other young men that are similar to their age. So we were able to statistically show that this is really what's going on. And since the first wave of the study, now there is a substantial reduction in new infection rates among those young women in sub-Saharan Africa. So that's something that I'm personally very proud of. And this is something that you can also do if you're doing our profession, if you come here.
Excellent. Well, thank you so much to all of our faculty panelists here. I would ask that if you have questions, they're going to be back in the lunchroom as well to answer further questions, have more conversations and discussions. They're looking forward to exploring these topics further with you. So thank you all for your time and for your expertise, and we really appreciate it.
Sure.
Thank you.
Thank you.